And so let me introduce you Rebecca Belletti. She is affiliated with the Universitat Politècnica de València, and she is presenting a work in collaboration with the colleagues from the same university and also the Georgia Institute of Technology. And specifically, her talk is dealing with the mutation in human atrial cells and their impact on arrhythmogenesis. Please, Rebecca, the stage is yours.
Okay, thank you very much. Good morning, afternoon, or good night to everybody. So to start, here's a quick overview on the structure of the presentation. And so we can move on with the motivation which is behind this work. As we may know, atrial fibrillation is the most common cardiac sustained arrhythmia, which affect more or less 1.52% of the general population. Usually, atrial fibrillation comes as a consequence of some pathologies as cardiovascular or endocrine disorder, or as a consequence of aging or lifestyle factors as high consumption of alcohol, smoking, or obesity. But sometimes atrial fibrillation manifests even in patients in which these risk factors are not present. This happens more or less 30% of the time, and this is the case we refer to as low atrial fibrillation. In low atrial fibrillation, over years have been demonstrated to be related to have an inheritability component and to be related to a genetic predisposition of the patient to have this fibrillatory event. So this means that there is a genetic mutation which affects genes that encodes for protein structure of ion channels. So in particular, in this work, the gene we are focusing on is the KCNH2, which is the gene encoding for the protein structure of the rapid delayed rectifier potassium current. We focus on this gene because the rapid rectifier potassium current is one of the main repolarizing current. So this means that if we have a genetic mutation affecting this gene, we probably have a abnormal conduction of the IKR current, which can lead to a shortening or lengthening of the action potential duration, which has been demonstrated to be more prone to atrial fibrillation. So the aim of this work is modeling and simulation of the effect of two gain-of-function mutations, which are T895M and T436M. So the experimental data of the two mutations were taken from literature and from a previous study in which the two mutations appear in two male patients, both affected by low atrial fibrillation, which then developed in persistent and paroxysmal atrial fibrillation. The formulation of the IKR current we choose is the one from Hodgkin-Huxley that then has been modified, adding some parameter in order to model the effect of the two mutations. Through a sensitivity analysis, we came up from 7 to 4 parameter that help the fitting of the mutant experimental data. Then this modified IKR formulation has been integrated into the Court-Munch human atrial single cell model in order to study the effect of the mutations in single cells simulations, and so on action potential, duration, and restitution properties. The parameter estimation procedure has been carried out using the genetic algorithm as optimization algorithm. Main parameter population size and generations were set at 100. So we have the generation of an initial population in which each individual has been evaluated through the evaluation of fitness function, and a score value has been associated to each individual. Then we actually enter the structure of the genetic algorithm in which through selection, individuals with good fitness function were selected as parents for the successive generations. Then crossover methods were used to mate the two parents, and this crossover happens in a randomly chosen site. And so in that site, the genes binary representation were swapped. And then also it has been applied a mutation operation on each gene of each individual with a mutation rate of 0.9. So these are the results from the fitting procedure. As we can see, to the left, we have results from the mutation T895M, and to the right, the results of the mutation T436M. In the tables, we have the combination of parameters which have been retrieved thanks to the genetic algorithm. And as we can see from the picture, these parameters were able to fit quite accurately the experimental data which here are indicated as target with their experimental ranges. Then we moved to single cell simulations. In the model, we added also the acetylcholine potassium current with an acetylcholine concentration of 0.005 micromolar. And the results shown are shown after 60 pulses of stabilization with a BCL of 1,000 milliseconds. So for right atrium, as we can see, in presence of the mutation T895M, the action potential shortens of 17% the wild type value. And this is due to a higher peak in the potassium current, which is 62% higher the control case. Also for the mutation T436M, we have a shortening in the action potential duration of about the 8%, with a higher IKR peak of which is 26% higher the wild type value. In both cases, we studied restitution properties. In particular, we noticed that for the mutation T895M, the restitution curve has a maximum slope value which is reduced respect to the wild type restitution curves, and this does not happen in case of the mutation T436M. In left atrium, also we observed in case of mutation T895M a shortening of the action potential duration of 23%. Because of a higher, of a 60% higher peak in the potassium current. In also the effect of the mutation T436M has been studied. Also in this case, action potential duration shortens of 12% with a peak in the IKR current of 26%. Restitution curves are shown and studied, and also in this case, we have a reduction of a 31% in the maximum slope value in case of the mutation T895M, but not in presence of the mutation T436M. So then we perform some 2D simulation on a two-dimensional geometrical mesh with dimension 5 by 5 centimeter square with a resolution of 300 micrometer. And well, the mesh is shown in the picture. So we apply 10 planar pulses to stabilize the tissue, and then an S1, S2 protocol. The S2 impulse was a rectangular stimulation in the lower left corner of the mesh, and then we perform also phase analysis in order to get the rotor steep trajectory. So the only case in which we observed a reentrant activity that can last more than 5 seconds of simulation was the case of left atrium and in presence of the mutation T895M. So let me see. I wanted to show you the video of the reentry. And as we can see, mostly from the phase map, the reentry occupy a very large area, but the pattern seems almost regular over time. But of course, further studies needs to be done in order to assess the effect of the two mutation in 2D simulations. So in conclusion, we can say that we were able to characterize the effect of the two mutations both from the channel dynamics point of view and at single cell levels. We have observed that the action potential duration at 90% of repolarization shortens in presence of both mutation, but in presence of the mutation T895M, this shortening is more evident, and this effect is even more strong in case of left atrium than in right atrium. We also observed that the maximum slope value in the restitution curve decreased in presence of the same mutation, the T895M, but this does not happen in case of the mutation T436M. So the resistance to high frequency pacing excitation and the action potential shortening and also effective refractory period shortening can just lead us to think that the two mutation can generate a substrate that can be more prone to lead to an atrial fibrillation episode. And this has been also demonstrated with the 2D simulation, even if it happens just in one of the cases studied. Of course, more tissue simulation have to be done, and also we have to move on with wall atria and torso simulation in order to better understand and characterize the effect of this mutation, and in order also to go on with a patient-specific approach. Well, we also are working on the definition of a methodology that can help in the cardiac reparameterization problem, starting from the genetic algorithm I presented and providing different selection, crossover, and mutation method in order to obtain a better reparameterization in cardiac problems. Thanks for your attention.
Thank you, Rebecca, for the interesting talk. There is time for questions, so if you have comments or questions, please raise your hand or write me in the chat. Okay, well, let me start. My question is rather technical. At the beginning of your talk, you mentioned that you performed some sensitivity analysis. By which tools you performed it, or if you proceed by trial and error, if you envision the possibility of using, let's say, more sophisticated tools or automatic tools for performing it.
Yes, I've basically changed the value of the parameter in a wide range of value to see which are the effects of these parameters on the curve we were interested in.
Okay, But I mean, you foresee using, let's say, some numerical method specifically aimed at this, or you're satisfied with the output and the cost that is involved in this study?
I think we had a good at the end. We were able to find a less number of parameters respect to the one we started with that were able to produce a good fitting of the mutant experimental data. So I would say that I'm quite satisfied with the fitting that the parameters provide.
Okay, thank you. Meanwhile, we had a question in the chat by Gary Mirans. Did you try to predict some current recordings for a voltage clamp protocol that was not used in the fitting for the two mutants?
Oh well, no. Actually, we had we applied the voltage clamp protocol to obtain the synthetic current as shown in the literature.
Okay. Meanwhile, we have another question from Professor Dossel. Please, can you turn on your mic?
My question is related to that. Thank you, Rebecca, for this interesting talk. It's related to the last question. You optimize these parameters P1, P5, P6, P7 for the two mutants. How sure can you be that this is the only set of parameters that gives data that resemble the experiment? Could it happen that another set of parameters, P1, P5, P6, P7, has the same quality but the different parameters?
Yeah. Well, aside from the sensitivity analysis, we were not sure if these parameters were actually the ones that can have that can lead to a better, let's say, the best reparameterization of this mutant experimental data. But then we tried also to use some other parameters that can have a similar effect, but we came up with this set of parameters that, of course, are extremely related to the experimental data we had.
Mm-hmm. Did you start your genetic algorithm with a large variety of initial values?
Yes.
Okay, then. Okay, thank you.
Okay, thank you, Professor Dossel, for your comments and questions. And if there are no further questions, then we'll move on. Let me thank, first of all, Rebecca for his interesting talk.